An in vivo CAR-T company just posted first human lymphoma data, and the headline is almost offensively clean. 100% response rate, 83.3% complete response rate, no lymphodepletion, no serious adverse events, and no ICANS. That is big. It is also 6 patients at dose level 2, with a median follow-up of 2.2 months, after the lower dose looked nowhere near that strong. So yes, pay attention. No, do not lose your adult supervision. This is a claim check on Legend Biotech's LB2501 update announced on June 2, 2026, ahead of EHA 2026. The goal is not to do the usual biotech thing where we hear 100% and immediately begin levitating. The goal is to translate what actually changed, why it matters, what people are missing, what could still fail, and what to watch next.
Start with the actual dataset. In the abstract filed with Legend's 6K, 12 patients with relapsed or refractory B-cell non-Hodgkin lymphoma had been dosed as of April 1, 2026. That included 7 large B-cell lymphoma patients, 3 follicular lymphoma patients, and 2 mantle cell lymphoma patients. Across all 12, the objective response rate was 50%, and the complete response rate was 41.7%. But at dose level 2, the headline cohort, the objective response rate was 100%, the complete response rate was 83.3%, and all responses were ongoing at cutoff. That detail matters a lot. If your summary is just LB2501 got 100% responses, you are compressing away the entire dose-finding part of the story. Dose level 1 showed CAR-T expansion in 5 of 6 patients, and the overall trial-wide response rate is not 100%. The clean headline belongs to the higher dose cohort, not the full study. Why is this still a big deal? Because this is one of the clearest human signals yet that in vivo CAR-T can generate meaningful anti-tumor activity in B-cell malignancies without ex vivo manufacturing and without lymphodepleting chemotherapy. That is not a mouse win. That is not a monkey win. That is not a slide with arrows and vibes. That is an actual human oncology readout, and the field needs more of those, more than it needs prettier platform diagrams. The pharmacokinetics are part of the story too. Legend reported dose-dependent in vivo CAR-T expansion, with detectable CAR-T cells in peripheral blood for up to 116 days. At dose level 2, the median Cmax was dramatically higher than dose level 1, and the median Tmax was 15 days. Translation: the higher dose did not just look better clinically, it also looked more biologically real in the blood. Now, the safety card. No dose-limiting toxicities, no serious adverse events, no deaths, no ICANS. Infusion-related reactions happened in 75% of patients, all grade 1 to 2. CRS happened in 66.7%, again, all grade 1 to 2, and no patient needed tocilizumab or glucocorticoids, according to the abstract. That is encouraging. It is also early. Small cohorts are where you learn a signal exists, not where you declare the safety argument finished forever. Here is what I think people are missing. This is not just a response rate story. It is a control story. Legend is using a third-generation replication-incompetent lentiviral vector with a modified fusion glycoprotein and a CD3 binder to bias transduction toward T cells. The abstract also says insertion site analysis looked highly T-cell specific, polyclonal, and mostly single copy per T-cell. That is exactly the kind of boring control detail that decides whether in vivo CAR-T becomes a therapy class or just a very expensive PowerPoint genre. But this is also where you should keep your caveats alive. The follow-up in the hot cohort is short. The median for dose level 2 is 2.2 months, with a range of 2.0 to 3.8. The disease group is mixed. LBCL, FL, and MCL are not identical clinical problems, and this is a first-in-human phase 1 dose escalation study. If you hear all that and still say basically solved, congratulations, you have been successfully marketed to. This is why I think the right viewer filter is simple. When an in vivo CAR-T company posts a beautiful early number, ask 3 questions. First, is that number for the whole study or the best-looking cohort? Second, how long have those responses actually been followed? Third, what control evidence do they have that the right cells got programmed in the right way?
If the answer to any of those gets blurry, the headline is ahead of the biology.
The broader field context matters too. A recent Nature Review's drug discovery review argued that in vivo CAR-T is trying to escape the cost, logistics, and waiting time pain of ex vivo CAR-T, but it inherits new questions around targeting specificity, durability, host immune responses, and regulatory oversight. In plain English, factory-free is not failure-free. You can absolutely make access easier and still lose on biology, persistence, or safety. What changed here versus the earlier Kelonia-style conversation in myeloma is disease context and proof texture. LB2501 is pushing into B-cell NHL. It is dual targeting CD19 and CD20, and it is showing that the human category signal is not isolated to one disease thesis. That matters. But the category fight is still wide open. Lentiviral delivery, targeted LNPs, fusogens, selective translation, site-specific integration, all of that is still competing for the control stack that survives contact with real patients.
What could fail from here? Durability could soften as follow-up matures. Later patients could behave differently from the first 6 at dose level 2. Safety could widen outside the earliest cohort. Mixed indication performance could end up uneven. And even if response holds, the field still has to answer the late, uncomfortable questions around repeat dosing, immunogenicity, long-tail safety, and whether convenient delivery actually translates into durable competitive outcomes. What should you watch next? Watch for more patients at dose level 2. Watch whether complete responses remain deep after 6 months and beyond. Watch subtype-specific breakdowns instead of pooled optimism. Watch whether investigators keep seeing T-cell-specific transduction when the dataset gets bigger. And watch whether anyone can reproduce this kind of human signal with a different delivery stack, because that is when the field stops being a curiosity and starts being a real platform war.
My opinionated wrap-up is this. Legend's LB2501 data are real and important. This is not fake progress. But the practical conclusion is not 100% means solved. The practical conclusion is that in vivo CAR-T just earned a more serious class of question. The headline is a gorgeous dose level 2 response rate. The real story is whether that signal survives time, scale, subtype messiness, and safety.